This is the beginning of our nervous system unit. We're talking about nervous tissue. So we'll begin with nervous tissue and eventually we'll work our way up to the brain, cranial nerves, spinal cord, spinal nerves, senses, autonomic nervous system. It's a big unit. We've gone over all the other tissue types, epithelium, connective tissue, muscle, a little bit at least. There's more to cover for those. But now we're focusing more on nervous tissue. So we'll be distinguishing the different types of, well, the nervous tissue as a whole, and also the different divisions of the nervous system. The specific neurons and things called glial cells within that is also part of nervous tissue that make up the nervous system. We'll look at the anatomy of a neuron and how the shape of a neuron helps with its function, how it can sense, integrate, and communicate. We'll also understand the different parts of reflex pathways. What is a reflex and how does that work? Histology will be really important for this chapter. So we'll be looking at the different shapes of neurons and also of nerves and connective tissues and special sensory receptors. So there's a lot in this chapter. This is going to be covered over, I believe, 5 videos, this particular chapter. Then there's more chapters relating to the nervous system. When you think of reflex, maybe think of the knee-jerk reflex. The knee-jerk reflex, you hit your knee, you're supposed to kick your leg out, and you kick your leg out. So what's going on is that there's a stimulus. You're hitting your knee. That's the stimulus. You have to somehow, your body has to somehow know that you're being hit. So you sense being hit, it stretches something out. We'll get to what exactly later. And then that information is sent to your spinal cord. So imagine the spinal cord right here. Something goes on in your spinal cord where you are deciding what to do with that information. You sense that you've been hit. What do you do in response? That decision-making process, the word we give for this is integration. So we go from sensation, sense that we've been hit, to integration, we're deciding what to do with that information. And then we do something. We send a signal to the muscles of our leg, and it causes our leg to kick out. That is our motor output. It doesn't have to be motor, so let me actually remove motor. But this is our output. Sensation would be input. Something comes in, you decide what to do, and then you have your output, your action. So as you can see, there's a lot going on in this reflex, and we have to see what goes on at the sensory side. We have to see what goes on in the integration side. We have to see what goes on in the output action side. And neurons and glial cells are all part of this. Nervous tissue is all part of this. To remind you where we are, we've seen this slide before in the previous videos. Tissues are groups of cells that work together. They're from a similar origin. Since we're talking about nervous tissue, remember that comes from the same embryonic tissue as epithelial tissue. It comes from ectoderm. Since these cells, these tissues are so small, we need to study them under the microscope. That's what histology is. We'll be seeing lots of histology in this chapter and the coming chapters. They carry out a similar function. There's interstitial fluid, and we'll see the importance of what goes on outside of these cells as well. We're focusing on nervous tissue, one of the 4 major tissue types. Nervous tissue, what sets this apart from the others is that it not only has electricity that spreads through it. Remember muscle? Muscle has electricity spreading through it. So does nervous tissue. The difference is that it controls other things. It can communicate with other things. Control and communicate. That's the major thing going on here. We can sense what's going on, we can decide what to do, and then do something about it. We can control muscles, we can control epithelial tissue, we can control what connective tissue does. controls many different aspects of our body. Not everything, but many things. When you think of nervous tissue, you probably think of neurons. Neurons are the major cell. When I say major, I mean they're the functionally important cell type of the nervous system. They're specialized cells that process and transfer information. They communicate. We've seen pictures of them before. Here's a basic illustration. Here's a scanning electron micrograph. You can see the cell body and its branches. By having these branches, it can better communicate with different things, receiving input or sending output. Since the nervous system is a control system, we can use neurons to send quick, short-term responses. For example, let's say I want to move my finger. A signal from my brain via neurons gets sent from my brain to the muscles of my finger. I cause contraction of my muscle and I do it once. If I want to do it again, I have to send another signal and then another signal and then another signal. But it's quick, milliseconds long, and it's short term. It just does it once and that's it, unless I keep sending signals. Compare this with the endocrine system. Let's say you release testosterone in your body, high amounts of testosterone. That high amounts of testosterone causes changes in your body. It communicates your body to, for example, produce more hair. The hair is going to be there long term. I mean, you can lose it, you can shave it, but it's still that long term on the scale of minutes to hours to days where that hair starts growing. That's a long term, slower response. Slower meaning more than milliseconds. Nervous system is very quick because it relies on electricity. Electricity spreads through these neurons, and these neurons some are very short, but some can be very long, spreading an electrical signal quickly through our body. It's just like muscle fibers, you're spreading this electrical impulse along the plasma membrane. We're going to see a similar mechanism to what we saw with the mechanism for spreading electricity, say down T tubules. Neurons have a very long lifespan. Some neurons you keep your entire life. You may hear that you can't make new brain cells. That's not entirely true. You can. There are some parts of your brain where you grow new neurons. We're still trying to better understand what those new neurons do. But most neurons live very long, and most cannot undergo mitosis. So there are some that if you do damage, we don't have the capability at this time to grow new ones. Our medical technologies not where we would want it to be. Say if you damage your spinal cord, you can become paralyzed, and we have no way at the moment to regrow those neurons, repair those neurons. Because these neurons are working constantly, because they're sending electrical signals, to do that, that takes energy. They have a very high metabolic rate. Metabolic, that word means, it means energy usage. These cells use a lot of energy. This is an anatomy class, not a thermodynamics class. So what is related to metabolic rate? What cellular structure? These cells have lots of, these cells have lots of mitochondria. Within their cells. Have a high amount of mitochondria within their cells. So what we see here, let me go back here. What we see here is a generic looking neuron. You see a generic looking neuron here. Some neurons look like this. Not every neuron looks like this. We're going to go with this look for now, and then we'll introduce different shapes in a minute. So here's a single, well, here's actually two neurons. There's one neuron right here, and then there's a start of another neuron going this way. The way neurons basically work is that you have one end, generally, one end that is for detecting a signal. One end is for detecting a signal, stimulus, something communicating with it. And then another end that is for sending signal out. So one is for input, one is for output. The input side, those are dendrites. Dendrites are these branches. Dendrites are these branches that receive signals. So what I've outlined here are dendrites. I'll also outline the dendrites of this neuron here. It's a different one. And you can see that it's going to receive input at this location. Let me circle the location. It's going to receive input at this location and at this location in what we've drawn here. Because this other neuron is going to send some kind of signal to it. You could be getting a signal from another neuron. It could be a signal from the outside, could be something from within you, like knowing how stretched out your stomach is, or the amount of carbon dioxide in your blood. Many different things that we can detect. But dendrites detect it. The word dendro, the root word dendro, means branched. Remember Langerhans cells? Langerhans cells were also called dendritic cells because they're very branched. So dendro just means branch. These are branches coming off of the main cell body. This is the cell body of the neuron, the main part where you find the nucleus and most of the organelles. There's one region extending off from the cell body into a really long extension. Typically very long. Again, it can vary how long this is. But the really long extension, the term for the long extension is the axon. And axons send signals out. They announce. You have an electrical signal spreading down in one direction. It doesn't go backwards. It only goes in one direction to the very end. The beginning of the axon, so the junction between the axon and the cell body, that's called the axon hillock. So this region right here is known as the axon hillock. The ends of the axons, we've actually seen this before because we had them communicate with muscles. The ends of these axons, those are called axon terminals. So you have axon hillock at the beginning, the long axon, and then axon terminals. And there can be many because the axon can branch. Axons announce, they send out a signal to another neuron or to a muscle or to a gland. They're telling something what to do. They're communicating. We're not going to go over this quite yet, but I want to point out some things on this particular axon. Not all axons have this, but you can see that there's this covering called a myelin sheath. It's covering the axon in segments. Think of it as an insulation. What are you insulating? We'll get to that. But these segmental coverings are called myelin sheaths. And then because there are segments, there are gaps. And those gaps, those are called nodes of Ranvier. So each one is a node, plural nodes, named after a French guy, nodes of Ranvier. Dendrites are for detecting. They recognize the input signal. The cell body and axon hillock, they're for integrating. They're for your decision making. The decision, what is the decision? It's not, when I say decision, it doesn't have to be something super complex, like deciding what to wear today. The decision is, should I send a signal out or should I not? It's a binary, send a signal or don't send a signal. That's what's going on in the cell body and at the axon hillock. And we'll see what structures relate to that decision making. There is anatomy in this. When you send a signal out, that's the axon. The axon That is our output. You're going to see me mention this over and over and over again. Whenever we have a nervous system reflex, there's an input, integration, and output. We have it at the cellular level. We'll see it at the whole system level, that there's integration, or input, integration, and output. Let me clear this for a second. I want to point out that when an axon terminal communicates with, say, a dendrite of another neuron, there's a gap of I shouldn't call it gap because I don't want it to be the same thing as those other gaps, but it's this interstitial space between the axon terminal of one neuron and the dendrite of the next neuron. That space is called the synaptic cleft. Cleft means like to cut in half, like a split part. Together, the axon terminal, the synaptic cleft, and dendrite or whatever is there on the opposite side, together, that's called a synapse. A synapse is this whole region here. And there's another synapse right here. Synapse is an area of communication. We've seen synapses before. We saw a synapse with a neuromuscular junction. A neuromuscular junction is a special kind of synapse where a neuron controls a muscle. You have an axon terminal of a neuron communicating with the motor end plate of a muscle. We could have a synapse with one neuron controlling another neuron, so that we get this relay of communication. We can have a neuron control glands. So neurons can control when a gland secretes something. Not all glands secrete this way, but some glands do. Point is, neurons can form synapses with other neurons, with other muscle fibers, with glands. They can communicate with many different things and therefore control many different things. Now, neurons are functionally the most interesting part of the nervous system, but they're actually outnumbered by a different cell type that is also considered nervous tissue. This is called a glial cell. The word glia in German means glue. I believe it's German. So it's like the glue, it's not actually glue, but it's kind of supporting. That's what we mean by the glue. It's supporting and protecting neurons. They're the support staff. Neurons are these large cells that branch and can be super long. These glial cells are these tinier ones that surround or cover and protect neurons in different ways. How they support and protect, let's take a look. And just to show you some size differences, take a look at this histology at the bottom right. You can see the cell body of this neuron. That's the cell body. You can see the axon, hillock, and axon. And then all these ones I'm going to color in dark blue. These are glial cells, very small compared to the neuron cell body. They don't branch quite like this. They're not as big. Very, very different structure and therefore very different function. So there are several different glial cell types. One glial cell type, or I should say two different glial cells can be found, two different glial cell types can be found in what's called the peripheral nervous system. I have to define what peripheral versus central is. The central nervous system is your brain and spinal cord. The central nervous system is your brain and spinal cord. Everything else, the nerves that branch off of your brain and spinal cord, the nerves plus cells. I haven't defined what nerves are yet either, so we'll have to get to that. Anything that's outside of the brain and spinal cord, that's peripheral nervous system. So if you have neurons outside of the central nervous system, outside of the brain and spinal cord, in the peripheral nervous system, the glial cells that you can find supporting these neurons, there's two different kinds. One is called a Schwann cell, and the other is called a satellite cell. A Schwann cell, Schwann cells, what they do, they wrap around axons. They wrap around the axons of neurons to form those myelin sheaths. Let's wrap over and over and over again. You can see those layers in this picture. In this picture at the bottom right, this histology, this is a cross-section of an axon. So all those dark dots you see, are axons. And then the white part that's surrounding is the myelin. And so that white part, therefore, that's the Schwann cell in this example. Schwann cells cover axons to form myelin. Satellite cells are a different type. Take a look at this histology over here. I'm going to circle a neuron. Here's one neuron. Here's another neuron. Here's another neuron. Here's another neuron. Here's another neuron. They don't look branched. They are. You just can't see the branches. But there's the cell body of the neuron. The cells that are surrounding here, 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 here. These cells, those are satellite cells. Like a satellite going around the Earth, it's just orbiting around. It's surrounding. Satellite cells, they're your basic support cell. They provide nutrients, they help clean up wastes. They do all these basic support type role for the neurons. They're there to help support the neuron cell body. So satellite cells are around the cell body, while Schwann cells are around the axons in the peripheral nervous system. I suppose I'll say, what is the function of the myelin sheath at this point? When we cover and wrap an axon, what we do is we insulate it. We insulate the electrical current. Electricity can be lost. It can spread outward away from your main path. When we have electrical wires, we cover it, we insulate our electrical wires with rubber or some other insulator so that electricity doesn't shock us and it doesn't lose as it travels from one point to another. By covering our axons with myelin sheaths, in this case formed by Schwann cells, we're insulating the electricity so that it spreads faster. Myelin sheaths spread electricity faster for neurons. You get faster conductivity. In the central nervous system, so here we are in the central nervous system, and once again, this means brain and spinal cord. We have different glial cells. So those two that I just mentioned, the Schwann cells and satellite cells, they're only found in the peripheral nervous system. These ones are only found in the central nervous system, in the brain and spinal cord. Let me go over this one here. Oh, no, sorry. Let's just go over this one first. This one here on the left, this is called an ependymal cell. If you look at the histology, it looks a lot like a epithelial cell. It's lining a lumen. Do you see these cells lining this space? These cells line compartments within the brain and spinal cord. If you're unfamiliar, there are some regions within the brain and spinal cord filled with a fluid called cerebrospinal fluid. Think of cerebrospinal fluid as a super highly filtered blood. There's no cells in the fluid, it's just filtered, and you have nutrients, and you also get rid of waste in that space too. So ependymal cells line these fluid-filled spaces of the brain and spinal cord. They line ventricles, and they line something called the central canal of the spinal cord, not the central canal of bone, central canal of the spinal cord. They also produce and help circulate that cerebrospinal fluid. So they're the ones, if there's a blood vessel right here, let's say there's a blood vessel here. If there's, let's say there's glucose here in the blood, glucose can exit the blood, exit the plasma, go inside of these ependymal cells and get filtered out into one of these spaces to form cerebrospinal fluid, along with water and other things. You're preventing bad things, you're trying to at least, you're trying to prevent like toxins or pathogens that might be in your blood from getting into your cerebrospinal fluid. You don't want to expose your brain and spinal cord to those type of things. It's possible for that to happen. We'll talk about infections like meningitis later on, but in general, that's what we're trying to protect. Those are ependymal cells. Another cell type, another glial cell type is called an astrocyte. Astro means star. If you're an astronaut, you go to the stars, you go to space. But these are star-shaped. They are branched, kind of like neurons, but these are much smaller. Look at the difference in this histology between the cell body of a neuron versus the cell body of an astrocyte. It does branch a lot, but it's quite different. By forming lots of these branches, they can also support neurons. Remember those satellite cells? Those satellite cells were just around neurons and just generally supported, provided nutrients, took away waste, other things that we're not going to discuss. That's what astrocytes do. They're just there to help support the neurons, very, very generally. They can also do one other very specific thing. They can form something called a blood-brain barrier. If there is, I'm actually going to clear this so I can show you on a previous slide. This is slide 9. You can see a blood vessel right here, and you can see extensions of the astrocyte covering the blood vessel. What these astrocytes do, this is only showing one astrocyte, but if you have another astrocyte here, another astrocyte here, and they completely cover up the blood vessel. You want to provide oxygen and nutrients to the brain and spinal cord, but you don't want to let anything in the blood get to your brain and spinal cord. That's how that could lead to infection, and we don't want to infect our brain and spinal cord. It's super vital that we keep those functioning at a very high, we don't want them to get hurt. We don't want them to get sick. So we cover up our blood vessels with astrocytes so we prevent just anything from escaping. We want to regulate where things come in using our ependymal cells. So by covering up blood vessels with astrocytes, we're regulating what's actually coming in and contacting most of our brain and spinal cord. That's what the blood-brain barrier is. Astrocytes form the blood-brain barrier. They physically cover up blood vessels. They form tight junctions. They form tight junctions to prevent anything from leaking out from the blood vessels. There's one modified glial cell. It's actually an immune cell. And this is called microglia. Not the most creative name here. Micro means small. Glia, it's a supportive cell. Microglia, they are phagocytic. You remember Langerhans cells, which are also phagocytic? They eat up things, they eat up debris, they eat up pathogens. They help defend our brain and spinal cord against anything that might be around. So general cleanup crew, and also to help defend ourselves against potential pathogens. Finally, we have one cell type called an oligodendrocyte. Oligo means a few. Dendro means branches. So oligodendro means a few branches. It's a cell with a few branches. So you have the cell. You have the cell, here's the cell body. It extends a few branches, and each branch covers up axons. So this also forms myelin sheaths. So oligodendrocytes form myelin sheaths in the central nervous system, while Schwann cells form myelin sheaths in the peripheral nervous system. They do exactly the same thing in slightly different ways. Let me show you the difference. Schwann cells, take a look over here on the right. Each covering is a single cell. Do you see how the nucleus is off to the side? Each covering is a single cell, just wrapped over and over and over. You can see how the membrane wraps over and over and over, just covering up the axon. A single Schwann cell wraps one segment. With oligodendrocytes, though, you've got the cell off to the side, and you've got an extension wrapping around an axon, another extension wrapping around an axon, another extension, another extension. That's where the oligo comes in. A few branches wrapping around segments of axon. But in the end, they do the same thing. They form myelin sheaths, and they insulate, they prevent axons from losing as much electrical signal. They're insulating the electrical signal as it passes down the axon. In this histology, at the bottom, you can see an oligodendrocyte. You can't see the extension, but you see that it's not immediately next to the neuron because it'll extend out its branch. In this longitudinal section at the bottom left, what you see here, these are axons. And you can see a Schwann cell nucleus, and that would form little segments around each axon. If we look at the histology in the other videos, in the pre-recorded videos, we can take a look at slides where we have axons spread out, and you can see the spacing between the Schwann cells and along the axon. The gaps between the myelin, those are nodes of Ranvier, and we'll take a look at those. These nodes in the next video. All right, so we've gone over the basics of neuron anatomy and the different glial cell types, the ones we find in the peripheral nervous system versus the central nervous system. In the coming videos, we'll look at the different neuron shapes and how they relate to input, integration, and output. And then by the end, we'll put that all together and look at reflexes. So thanks. I'll see you in the next video. Please let me know if you have any questions.